Hi there, welcome to my channel where I share my science career job hunt experiences, deliver presentations on pharma, documentation, quality and microbiology, improve on my communication skills while helping you out. I am Paul Yeatman, a microbiologist of 15 years seeking a return to the lab. I can be found on LinkedIn, Twitter and my microbiology blog. If you are a subscriber, welcome back. Today I will talk about Welcome to Autoclave Validation, presented by me, Paul Yeatman, a Bachelor of Science graduate with over 15 years pharmaceutical microbiology experience. What is in it for you? Welcome to Autoclave Validation, presented by me, Paul Yeatman. Who am I? Well, I have 15 years experience in pharmaceutical microbiology, 9 years specifically with sterile injectables. 2 of those years I controlled the aseptic media fill program. I performed autoclave and incubator qualification and validations and lots of method validations. I have 6 years non-sterile manufacturing experience, and 8 years of all this is been in official supervisory roles, and outside of that I also try to lead anyway by sharing my knowledge and so forth via means such as this one. What is in this presentation for you? If you have no or minimal knowledge of the autoclave, well, this presentation will definitely be of help. If you have never audited an autoclave, then you will wind up with an idea of what to look for if and when you do. And if you are looking to get an autoclave replacing an existing unit or need to learn about validation and autoclave audit, then you'll get something out of this presentation as well. Life would not just be different for pharma if we didn't have autoclaves. Any manufacturer or laboratory requiring a sterilisation would need to be using radiation or chemicals or an oven to do their sterilisation. You still will find ovens used within laboratories to depyrogenate products, and also both within the lab and within production, formulated products are routinely filtered before being filled or filtered as a result of, say, testing. So what is the origin of this word autoclave? Well, auto comes from the Greek, and Latin is clavus for lock. So what happens practically is the pressure in the chamber increases, and there's a seal that, as the pressure goes up, that seal becomes tighter, which effectively automatically locks the device, hence autoclave. So what's some interesting info about the autoclave? Well, the man credited with initiating the project which led to the invention was Charles Chamberlain. He worked with Louis Pasteur, and he also invented a vaccine for chicken cholera, which was by accident. Some things in science are accidental, which have positive outcomes. This is one of them. If you want to look into the history of the autoclave, there's a whole bunch of other stuff that came before the actual thing we know as an autoclave, which could be sort of like an autoclave, but not quite. So I encourage you to look that up. What are the aims of this presentation? I will go through what an autoclave is, how an autoclave works and what may affect sterilisation. Why would you want to use an autoclave in either a production or a laboratory setting? I will identify the 2 main types of autoclaves used primarily in the pharmaceutical manufacturing industry. I will detail how to validate an autoclave, focusing mainly on performance qualification, which includes loading patterns, and I will detail what to look for in an audit based on my own experience and observations. The main focus of this presentation is the use of autoclaves in biological sciences, specifically the pharmaceutical industry. The underlying concepts can be applied further afield, so if this doesn't apply specifically to you, I encourage you to stay tuned and you'll still learn things. An autoclave is a device that uses elevated temperature and pressure in combination with moist heat to accomplish a task. So as we will discover, there's a lot of energy within steam. In biological sciences, this task is primarily to kill anything living. And due to that, it will also knock off viruses because the transfer of heat effectively denatures proteins, and that also includes things like DNA and RNA. And if they're non-functional, both living cells are now not living, and viruses can't do anything because they've got mangled RNA or even DNA, depending on what sort of virus they are. In chemical sciences, autoclaves are used to cure coatings, vulcanized rubber, for hydrothermal synthesis, and a lot of other stuff. So they're not just specific to a sterilisation use. Okay, the fun stuff. How does an autoclave work? Well, steam enters the chamber jacket, so we've got a little steam pipe here, comes in. As the steam enters the autoclave, it pushes the air out. There's a pressure regulator that maintains the jacket and the chamber pressure at a minimum of 15 pounds per square inch, which is the pressure required for steam to reach 121 degrees C, and the overpressure protection is provided by a safety valve. So as I talked about in an earlier slide, once the steam comes in, there's a seal, and the increase in pressure locks the seal down, which effectively seals the unit automatically. And after the autoclave cycle, everything gets evacuated, so the steam goes out, air comes back in, and then you'll do stuff like check temperature traces and whatnot to determine whether or not their cycle was actually valid, which I'll explain why that must be done later. How does an autoclave sterilise? So why wouldn't we just use soap or detergent and water to sterilise things? Well, that's only sufficient for sanitisation, which is the reduction of microbes, not the complete eradication of viable organisms. So soap or detergent and water is quite suitable for washing your dishes at home or washing bench tops, things like that, because like things you stick in your mouth, if there's low bioload, your stomach acid's going to knock off quite a large number of the microbes, which means that you don't wind up infected. Whereas you'd use an autoclave to totally eradicate any living organisms, because the equipment you're using or the products that you're sterilising, they can't contain anything living in them. Because if you were to inject, say, a terminally sterilised product into somebody that had still living organisms in it, that could possibly infect the person, give them septicemia, kill them. Now that's worst case scenario, which you don't want. And how does the autoclave work effectively? Well, steam held at the elevated temperature and pressure over time is used. That's based on D values, which I'll cover. Steam has a much greater heat capacity than boiling water. It's about 80 calories of boiled water versus 540 to produce steam. And at 1 atmosphere, that's a lot of energy needed to produce steam versus needed to keep get water up to the boil. And that means there's a lot of energy sitting there in steam, which could be transferred to proteins, which will denature them and render them ineffective. So yes, denature your proteins, because your proteins don't work, they're not biologically active, which means that whatever they're in isn't biologically active anymore. And if something isn't biologically active, it's dead. Note that autoclaving will not destroy endotoxins or pyrogens, which you may have heard of. What these things are is when they're injected into your bloodstream, they produce a fever. And it's also bad for patients, either immunocompromised sick ones or healthy people. When you get a fever, you tend not to be particularly well. If someone's sort of bad medically in the first place, giving them a fever is going to make them even worse. Again and again, you get the worst case situation of a dead patient, which could come back to haunt your production process. The type of autoclave you use will be dependent on your needs. So for production, you want to sterilise stoppers, bowls, tweezers, metal machine parts, caps, etc. Post-batch, you would want to use another autoclave to terminally sterilise product, possibly. This depends on, of course, your manufacturing procedure. You might be making sterile product or filling sterile product, or you might be making, say, not sterilised. You might be making product aseptically, so it's not sterilised, but the terminally sterilised product at the end makes it sterilised. And if you're a terminally sterilising product, ideally you want to have an autoclave large enough to fit your entire batch, so there's no deviations or discrepancies between cycle 1 and cycle 2, things like that. It's important to note here that if the autoclave is faulty or hasn't been validated within a certain amount of time and those cycles post-validation are no good, there's a risk to the end user. For microbiology, the autoclave has multiple roles, so you could be using the autoclave to sterilise equipment into the sterile testing suite. You could be using it for media making, decontamination cycles, and for preparing equipment for environmental monitoring. So if you're using an autoclave to sterilise equipment into the sterile testing suite, you could have like a front and a rear door. Alternatively, you could just have one door, and once the item's been sterilised, they could be put into a airlock or a pass-through for the testing suite. So it all depending on whatever procedures you have and how things are set up in the lab. For environmental monitoring, it is good practice to double wrap equipment. What would happen here is the person conducting the monitoring would take off the first layer of the autoclave wrap in the production airlock and then take off the second layer before entering into the sterile core. And you possibly may have encountered benchtop autoclaves which are really small, and I'd be surprised if you haven't at least seen pictures of home pressure cookers which act on the principle of pressurization and steam to tenderize meat. So basically there what you're doing is you're denaturing the meat proteins, which makes the meat, I guess, nicer. But the underlying principle is the same. There's temperature and pressure for a certain amount of time, does the job. So the production autoclave, it's usually big because you've got to autoclave a lot of stuff that goes into the production area. You'd load it up one side, say in your mixing room or your container prep room, and unload in the other side, which would be like your class 100 or class 10,000 zone, depending on your setup. Use to sterilise production equipment. It might be used to sterilise filled product after a batch, so in that case you could have one opening if you wanted. So you load it up within the filling room, unload it in the filling room. Alternately, you can load up in the filling room and unload it on the other side, like in your mixing room or filled product zone. If faulty, this has a potential impact on the sterile core if it's sanitising things into the room, or could affect batch disposition if it's a terminally sterilised function. For the microbiology autoclave, they may be large or small, all depends on your needs and what your URS, which hopefully was written before you got the unit, specifies your needs and wants. These autoclaves usually load and unload from the same side. Sterilised items do not unload directly into a production environment. They might unload into your sterile testing suite, as discussed. They use to sterilise equipment as well as media, and also use to decontaminate materials before disposal using what's generically called a decon cycle. Why use an autoclave? Well, it's the easiest way to sterilise large volumes of heat-tolerant materials. So I'll discuss why it's better than dry heat and chemicals and radiation. Why is an autoclave better than dry heat? Steam penetrates better than heat radiation, and it's also faster at killing than dry heat. Steam also is not as high a temperature as dry heat, and it won't burn many substances despite having a lot more thermal load than hot air. For example, you can sterilise filters in an autoclave. Using a dry heat oven would result in char. If you've got access to a sauna, jump in the sauna when there's no water being put onto the coals, and it might be really hot, but it's bearable. as soon as you put the water onto the coals and you get water vapour in the room, unless you've got really great heat tolerance, you'll find it very uncomfortable at that point. So that just shows you how more effective it is having moisture in the air versus no moisture to actually transfer heat. Autoclaving is better than chemicals, as in chemicals that are toxic, because water is a chemical. You would need large volumes of chemicals, and they would likely contaminate what you were sterilising, and you would need to extensively clean up after use. This equals time and money, and that can be expensive. There are also disposable problems, which again cost time and money. And steam evaporates quite quickly, and any residue converts to water or water vapour, which makes up clean up pretty much automatic for an autoclave. So once you've run your autoclave cycle and you've checked the temperature traces and pressure traces to confirm that the cycle is actually valid, unload the autoclave, put the items in the resting area, and even though the wrap might be moist or wet at this point, give it 5-10 minutes and it'll be dry. Why is an autoclave better than radiation? Well, with radiation you need shields so you don't irradiate the surrounding areas, be that equipment or people or whatever else you might want to actually nuke. Radiation can change the substances being sterilised as well as kill microbes, so it can make glass coloured or make rubber hoses brittle. You might find that sort of effect when you do autoclaving, but the actual amount of time it takes to turn a hose from clearish to dark brown is a lot longer than if you had irradiated it. Okay, next point is, once validated, simple indicators are used to tell autoclave and non-autoclave material apart, but they're not used to actually tell you that the autoclave did its thing. You need to look at the temperature and time and pressure trace to do that. And why do you want to actually do that in the first place? Well, PICS 009-14 Annex 1 Part 88 states that there should be clear means of differentiating products which have not been sterilised from those that have. So that's why we use the indicators. However, this autoclave tape, that won't show you if sterilisation actually occurred. So the tape's only to be used to show that an item has been put through an autoclave cycle. The temperature and pressure trace, and also time trace from the autoclave cycle, is what you must look at to determine if your cycle is effective. And an inadequate autoclave cycle can still produce a change in the autoclave tape, as I said. Once the cycles have been approved, then the contents can be released for use. So here I recommend that you don't even open the autoclave until the traces have been confirmed and you've signed them off as approved, which effectively approves that equipment for use, and then you can remove it from the autoclave. What may affect sterilisation? Well, sterilisation is dependent on a number of factors. The initial bioload, the microbe sensitivity to heat, the time the autoclave is held at the sterilising temperature, the ability of steam to penetrate the items being sterilised, and with that, as steam is used to transfer the heat, tightly wrapped items or long tubing may not be properly penetrated, as would containers which are integral, say a shot bottle with a tied-on lid. That you can't get steam penetration in there, which means that the heat transfer isn't as good, which I'll discuss. So with the bioload, if you validate to kill 100 colony forming units, or CFU, and your initial bioload is 200 CFU, then your sterilisation will not be effective. If you validate using Micrococcus luteus, you'll get different results than when using Geobacillus stereothermophilus, as the microbes have different thermal survival characteristics. For example, typically we want a D-value of about 1.5, and Geobacillus stereothermophilus provides that. Whereas Micrococcus luteus won't have a D-value of, making something up, 0.1, just based on how quick it would die if you did expose it to an autoclave. And when you validate a cycle of 20 minutes at sterilisation temperature, running the sterilisation cycle for 10 minutes won't guarantee sterilisation. So you've got to make sure that if you validate for time equals X, that is the minimum time they actually do to validate the autoclave cycle for. Steam penetration. Well, steam needs to penetrate items in order to transfer its energy. So a bottle with a lid tightly screwed on won't allow steam to enter it. A long hose may not allow steam to get all the way to the centre of the hose. And tightly wrapped equipment may not allow steam to fully penetrate the wrap. And they also may not allow for complete circulation of steam around them. so incomplete penetration could occur. So in these cases, the only way you're going to get heat transfer is via radiation, and that's not as effective as moist heat. So there's the difference between, as we talked about on an earlier slide, putting something in an oven versus putting something under steam and pressure. And if you were to identify a worst case situation as part of this, which you do during your validation, then you've got to document it and show that sterilisation is effective here in order to be confident that your entire loading pattern is effective. So if you found that, I don't know, the back left corner of your autoclave was slightly cooler than the rest of it, you'd want to pay particular attention to that when you're doing your validation. What is validation? Well, validation shows that a process or item of equipment does what it is claimed to do. It's more critical for autoclaves used to support environmental monitoring than for media making or decontamination. For tasks such as media making, tests such as growth promotions or sterility testing will show up for media is substandard. Ensuring you use sterile equipment for microbiology tasks means there is less chance of you getting a positive result, and thus less chance of batches being placed on hold while action level investigation is conducted. Decontamination cycles are used to sterilise items that are known to be or are potentially contaminated. Such cycles are generally run at more than 2 times standard hold time and can utilise what is known as overkill. More on that later. And why would we even bother validating? Well, again, PICS 009-14 section 83 requires it. And we need to know that sterile equipment being taken into the sterile core, as in manufacturing, or your testing suite, is actually sterile. We don't want to contaminate production or our test samples. If we contaminate production, that could lead to an adulterated product. If we contaminate test samples, we could have false positives, which lead to aseptic manifold trials and delays in batch release and potentially batch scratching and things like that. And when we're doing our environmental monitoring, we want sterile equipment because, again, we don't want to introduce contamination into production, and we don't want to introduce contamination into the samples we're actually using to either collect samples or take back up to the lab as process samples to test. What comprises validation documentation? Pretty much the URS, which is the user requirement spec, written with nothing in particular in mind relating to brand or model. We've got an IQ, which confirms that everything matches the equipment we determined we needed in the URS, and when you actually get it into the site, is everything that was on the box in the box, and is the unit installed properly. Once the unit is installed, then we follow the OQ, which determines whether or not the item operates properly. For example, does the unit hold temperature and pressure correctly? And then once we've completed that work, we go into the PQ or performance qualification, which confirms everything in the OQ stands up when the equipment is actually in use. And all this should be covered by a validation protocol which sets out the procedures and also assigns responsibilities. And following all of this work, there is a validation report which you'd want to write, which details the results, make valid conclusions based on those results, and also documents failures and successes. And of course, if there was a failure with any of your testing, you want to fully investigate it and justify why the equipment has been approved for use if that's what you deem a valid conclusion to be. During your performance qualification, you want to determine what you can successfully sterilise with regards to what's in the autoclave, and also pay particular attention to the cold spots which you may have identified during the operational qualification. And depending on your setup, the validation department may be responsible for the bulk of this work. If you can do it yourself, I recommend it, as it's a great learning experience. So, getting on to the performance qualification, what is that? It's the validation of autoclave cycles and loading patterns. You want to determine things such as what sterility assurance level you need. For example, 10 to 6 is in the FDA guidance, so I encourage you to follow that for sterile products. If it's a medical device which isn't intended to be used on broken or compromised parts of the body, you might use a sterility assurance level of 10 to 3. Ideally, whatever the guidance says, that's what you want to do, as it's always great when you don't have to explain what you're doing to an auditor. So if you follow the guidance, then there's no question marks as to why you're doing something different. You want to consider the microbes D value. Typically we would use Geobacillus stereothermophilus, which is a spore farmer, and the D value of this microbe is 1 to 1.5. You want to consider whether you use physical, chemical or biological indicators. The physical indicators would be the thermocouples during the autoclave validation, and the biological indicators could be spore strips or spore solutions depending on the product you're testing. So when I've validated or revalidated autoclave loading patterns, I've used biological indicators in the form of strips for an autoclave cycle within the lab, and spore solutions when I have assisted production in validation of their cycles. So with the spore solutions, production would bring up bungs, and we inoculate the bungs with a known volume of spore solution, which has known volume of microbes. And once that's dried, of course, according to your validated hold time, those bungs get sent back down to production. They go through the autoclave cycles. The bungs get brought back up to the lab. You fish out the bung that's been inoculated, which you can tell because it's a different colour to the rest, or you've labelled it a certain way. And you whack those bungs into TSB and determine whether or not anything grows. Whereas with the loading pattern validation, you'd have a spore strip next to a thermocouple and show that for the validated time and temperature, that actually kills everything on the spore strip. So effectively shows you that you have a 10 to the 6 reduction. And with this D value of say 1.5, once you do the calculation, that means that it will take 10.5 minutes to reduce the number of living microbes down to 1. So if you used a 10 to the 12 reduction, that would represent an overkill cycle, which would equate to about 18 minutes for this 1.5 D value. And there is a specific calculation for this, which I encourage you to jump on the internet and have a look for. And we've got a couple of links here, or a few links here you can check out, which are always handy. So what is this D value? Well, that refers to the decimal reduction time, which is the time required at a certain temperature to kill 90% of the organisms being studied. So that's population reduction by log 1. So after your organism is reduced by 1D, only 10% of the original organisms remain, and dependent on the microbe and initial numbers. So 1.5 equals 1 if you left after 10.5 minutes. So you give another 10.5 minutes, it goes down a certain amount. Z value refers to the temperature change required to produce a 1 log reduction in D value. And then we have this thing called F0, which is the number of minutes to kill a specified number of microbes with a Z value of 10 degrees C at a temperature of 1221.5 degrees C. That's often confused with the time the tamer is held at elevated temperature and pressure, and in practice it's the same thing anyway. And what is this thing called overkill that I've mentioned a few times? Well, we use many more microbes than we would find in items typically autoclaved, which negates the need to test the sample for viral load before running the cycle. So we have these millions of microbes on a spore strip, whereas the routinely autoclave equipment might have, say, 1,000, maybe 10,000. Like, really worst case situation. Using a sterilisation time exceeding what is necessary to kill a large number of microbes negates the need to determine the D value of the microbe. And also for a decon cycle, if you validated 20 minutes for your normal routine loading pattern, putting media in there or dirty equipment and autoclaving it for 60 minutes should be a long enough time to kill everything off. Ideally, you would determine the D value yourself, but to do that you need what's known as a beer vessel, which is a biological indicator evaluation resostometer. And last thing I checked, this costs about 100,000 US for one of them, which can be outside the budget for most organisations. A spore supplier devalue along with robust validation is vital to demonstrate your autoclave's effectiveness. And when I first wrote about beer vessels in a report, apparently validation got a good laugh because I spelt beer like B-E-E-R as opposed to B-I-E-R. So fun times back in the dim dark history of my career. Why is it important to actually validate loading patterns? Well, as I previously said, PICS, PEO 9-14 requires you to do validation. But it's also important to determine that you do get proper steam penetration, and that might occur in all cases. So it's important to show what you put in autoclave comes out sterile, and I discussed in the earlier slide when I've used spore strips and when I use solutions. So with validation, you want to validate 3 successful runs for each loading pattern, which shows reproducibility. You'd place the biological indicators next to your worst case spots and place a thermocouple also next to the biological indicator but not touching, just so you can show that the BI got to the right temperature over the right amount of time. How often you requalify depends on the use of the autoclave. Typically that'll be stated in the relevant SOP. It could be 6, 12, 24 months, all based on risk. So how much risk are you willing to wear? If you didn't validate every, say, 12 months, you did every 24 months, potentially you'd be looking at 24 months worth of investigation into potential risk to batch or impact on batch. So you can get away with it. The 6 months would be best because there's less investigative run around than say 24 months. For micro autoclaves, that depends on use. So if you're using the autoclave as part of sterility testing, I would suggest you do the validation as often as production would validate their autoclaves, just so there's alignment. If it's used for environmental sampling, whatever you've determined through your validation master plan, and decon cycles typically flow on from the loading patterns. So if you can show your autoclave works for your typical loading patterns, then your decon cycles should be effective. And of course, once you've gone through the effort of validating the loading patterns, they should be here too. You validate the worst case, so you could put less in the autoclave than the worst case. So if you've got, say, 20 by 1 litre containers in there, you could put in 10 by 1 litre containers. But you wouldn't go 21 1 litre containers or more because that's greater thermal load than what you validated. So you can always go less. With terminal sterilisations, steam penetration won't occur if the containers are integral, which they should be. So you will need to place vials containing spore strips amongst the loading pattern to show nothing grows in the vials, and cycle validation is critical to show unadulterated product. So even 1 CFU in a vial could be disastrous if injected into a patient. Touching on spore strips versus solutions again. Spore strips are used when they can be placed in or next to a test item, so like with this terminal sterilisation, or next to, say, an air sampling head in a micro autoclave, versus spore solutions when we want to have spores penetrate an item or get into the tight places, say down a centre cavity or stopper, possibly into the interior of a tube. But keep in mind that you need to flush out all any solution in the tube post validation. If you can get a spore strip down there, then I'd do that, because you always chop it out of the tube afterwards. Touching on terminal sterilisation again, if you validate a load of 20 ml ampoules, that could be used to show that a load of 1 ml ampoules at the same total liquid volume can be sterilised, because there's smaller volumes of water, less thermal load per item. Ideally you'd want to validate both cycles, but you could get away with just the 20 ml ampoules, as long as you don't see the total volume. One thing you should note here is that when you do your validations runs, the autoclave should be at room temperature, which is the worst case thermal load transfer. So a cold autoclave has to heat up more than an autoclave that is warm. And a cold autoclave may impact the ability of the autoclave cycle to kill off your BIs versus running a cycle immediately after another cycle, for example, when the autoclave is up to room temperature. or close to it. When using a worst case load, you also need to consider the physical features of the load. So is the equipment liquid, solid, porous, or something else, for example? Different materials may require their own worst case determinations, so you might need to validate additional loading patterns. So that's up to you with your documentation and your risk assessments. And what would be a worst case sample? You've got to validate that specifically or pay specific attention to it. So a long tube, a cold spot, a large volume of liquid versus a solid chunk of metal. These could all be worst cases for your loading patterns, so you want to pay attention to those when you're doing your validation. Getting on to the fun stuff, once it's all validated, what are your audit considerations? So these are what I would consider the important considerations. Was validation conducted? And if it was conducted, was it prospective or retrospective? If the autoclave's new, then I'd be looking at prospective validation, which is IQ, AQ, PQ, potentially looking at ERS. If it was an old autoclave, say greater than 15 years of age, IQ and AQ have probably been grandfathered, but you want to make sure that the loading patterns are current. And also you'd want to look at, say, preventive maintenance programs. The risk assessment might determine that you don't need that, but it's always a good idea to do preventive maintenance, because then the equipment's not going to keel over at a most inopportune time. The auditor would also be looking for SOPs and OIs and leak rate test data, basically all the supporting documentation needed to run the autoclave. You'd be looking at expiry dates. Are they current? So when was the last time the autoclave was validated? Is that aligned with your validation master plan and your SOPs? Was anything autoclave post-expiry of your patents, and if so, what is the impact? So there should be an investigation into the impact of something that deviated from the established procedures. Your cycle time, is it sufficient for the tested D values? So if you've got a D value of 1.5, is the autoclave runtime longer than about 11 minutes? Typically you'd find your cycles are 20 minutes long, so typically the answer would be yes, but there's something you need to look at. Was validation of equipment within calibration? So here, there's no point doing a validation on the autoclave if you say your thermocouples haven't been calibrated or your biological indicators expired, say, 3 months ago. You could do counts on your indicators, which would extend the validation period, but that would be whether or not that was allowed by your internal procedures. Some sites allow it, some don't. But you would need to document it and show that your expired strips or solution haven't expired. Are vacuum cycles used appropriately? So, for example, if you're sterilising media, do you have a fast or slow exhaust? You don't want a fast exhaust on, say, a liquid sterilisation cycle, because you're going to wind up with nothing left in your containers. Things like that. You might also want to use vacuum when steam penetration might be difficult, such as long piping or wrapped items. You want to examine the coolest and warmest positions, which will be stated in a validation report. You want to make sure that they've been identified, and if they have been identified, then were they investigated? So do you have data that shows that these cold spots, not so much the warmer spots, but most of the cold spots, is it possible to sterilise equipment in these positions? If yes, brilliant. You want to examine the largest or bulkiest loading pattern. Was the validation acceptable, and is there anything not listed on the loading pattern present in the autoclave? So as part of your auditing, you'd want to look at the autoclave, say after a cycle has been completed, and with reference to the loading pattern, once the autoclave has been opened up, determine if anything is in the autoclave that's not in the loading pattern. If that is the case, then clearly the procedures aren't being followed, and that's a big red X, and that's something that needs to be addressed. Were there any deviations from the protocols? So during your validation, did something go wrong? If so, was it investigated? I really hope it was. And then, were conclusions valid and justified? That should be a big yes. A couple of things that are not quite on your audit scope of the autoclave specifically, but you want to look at in the grand scheme of production and autoclaves and everything, is can the site show that product that has been terminally sterilised has retained efficacy. So you filled your product, you've autoclaved it and killed off any, say, pharmacological effect. That's not good because the product doesn't do what it's advertised to do. So you want to make sure that your sterilisation doesn't actually affect the product. You could also check that the steam system is validated and that dye bathing or some other integrity checking occurs post-sterilisation. So again, you've gone to the trouble of filling your product and thermally sterilising it. You can't then immediately release the product because that sterilisation action might have compromised the vials or ampoules by producing cracks in them. So you dye bath it or do some other sort of integrity checking. With the dye bath, once the items have been tested, they'd all be visually examined, and if any had dye in them, then they get thrown out, and the rest of the samples should be integral, which then means that they could be released for use. And with the introduction of PEO in 9-14, there was a lot of reworking of the parametric or testing in place release chapter, so you might want to revisit that in your procedures with regards to your autoclave validation, assuming you do terminal sterilisation. So, now that I've discussed autoclave validation and auditing, I would hope you know what an autoclave is and how it works, why autoclaves are used in a manufacturing environment, and why they're better in the most part than using chemicals, radiation or an oven. The basics behind autoclave validation, so your URS, your IQ, your OQ, your PQ, and some general things to look for in an audit. I have a link to the PDF version of this presentation in the video notes, and you can always rewind to something you want to hear again in this actual presentation. I should note that me presenting this topic online doesn't mean that you can take it and adapt it to train your own staff. If you want to do that, then I strongly encourage you to either employ me to do that, or better still, give me a job in your lab, production or QA department so I can use my skills and knowledge to help you and your staff on a long-term permanent basis. Which is good for me, good for you, good for the end user. I hope you have found this presentation informative, and I'd like to thank you for your time. And next up, we just have some references. So in order of appearance, here's the references. I'll also put them into the video notes, so you should just be able to click on the link and then get to it. So the wonders of the internet, yay! Thanks for watching. If you have found this video interesting or informative, please subscribe, like and share. Got something to say, such as a request for a future video? Put it into the comments. This has been Paul Yeatman, scientist seeking career satisfaction. Kapla!